Good afternoon, everyone. Thank you so much for joining us today. This is the Tyler's Hope for Dystonia Cure webinar. We're going to be discussing research and treatment. First, I want to say thank you to our sponsor for this event, John Spence, who's helped us out tremendously. We really appreciate everything that they do for Tyler's Hope. For those of you that don't know very much about Tyler's Hope for Dystonia Cure Foundation was established in 2006. Excuse me one second. Our goal is to discover effective treatments and promote awareness and education for this devastating disorder. Dystonia is a curable disorder, as you know, with enough money, we can continue to fund research for necessary to find a cure and bring top researchers in the world together on a mission to find a cure. Some of our accomplishments include the Dystonia International Patient Registry. We funded the creation of the largest neurology dystonia fellowship in the country. We have the Tyler's Hope Center for Excellence, the Dystonia Center at the Fixell Institute, and we've funded millions of dollars in dystonia research. Our first speaker today is going to be Dr. Michael Oaken. He is the current chair of neurology and a professor and executive director for the Norman Fixell Institute for Neurological Diseases at the University of Florida Health College of Medicine. He is one of the co-founders of the Movement Disorder Program at the University of Florida. He served as the medical director for the Parkinson's Foundation since 2006 and the medical advisor for Tyler's Hope for Dystonia Cure since the beginning. He's also partnered with Dr. Gundis and Foote to develop the first generation of closed loop adaptive deep brain stimulation approaches. And we are so grateful that he's joining us today. Our second speaker is going to be Dr. Edgar Rodriguez. He has 20 years of experience in gene therapy space with a focus on diseases of the nervous system. He has engineered and developed gene-based therapies to prevent and reverse neurodegenerative processes caused by the accumulation of toxic mutant proteins. He is currently scientific advisor to Tyler's Hope and chief science advisor of biologics and co-founder of the early stage biotech companies developed molecular therapies for CNS disease. And he also holds an adjunct faculty appointment at the University of Florida. So without further ado, I would like to introduce to you Dr. Michael Oaken.
Great. Well, thank you so much, Heather, and thank you to Rick Staub and Michelle Staub, the Staub family, Tyler's Hope, for having such a great foundation that's done so much good in the field and partnering with anyone and everyone to try to do good things for dystonia. And really what we're going to do today is, I think, have a chat. And so we have Edgar Rodriguez with us, who is an awesome researcher, and we've got the chat box open so we can live chat. We can talk about anything that you want to talk about, and I know I have a few questions that I'd like to ask Edgar about dystonia and DYT1 and the future and where things are going. And we also want to acknowledge that these are surreal times. You know, there was a great recent cast by the Dystonia Medical Research Foundation with Joel Perlmutter, who is their awesome medical director, talking about dystonia in the time of COVID. And it really is a very difficult time for a lot of people with diseases like dystonia, and being caught at home and getting cabin fever, and it can affect your symptoms, and it can be really scary. And so we've got the chat boxes open here. We're happy to answer questions on anything and everything. You know, we probably can do pretty well on a few sports, maybe baseball, but not all sports. And we can do some good on history and whatever else you're good at, Edgar, we can take questions on. But really anything that's on your mind, we're happy to answer. Now, just in in a larger 50,000 foot view, Tyler's Hope is a foundation that is really focused around DYT1 dystonia. And those letters DYT1, they indicate the gene that's abnormal. And this was sort of the first biggest gene that was discovered in dystonia. And it's not the only gene that causes dystonia, but certainly one that that has, you know, really been important to our development of an understanding. And I think if we're going to drive toward meaningful treatments for dystonias, we're going to need to really understand DYT1. And so, you know, this is a chromosome 9 abnormality. It's a deletion of a couple of pieces of the DNA. So it would be those little letters GAG. You know, if you were looking, you know, that deeply into the genetic code, and you know, that little deletion on the 9th chromosome causes all sorts of havoc and wreaks all sorts of havoc and can lead to symptoms for patients. And we know that you can get co-contractions where when one muscle contracts, another should relax. And if that happens at the same time, you can end up in abnormal postures. You can even get it in your voice box. You can get it in your face. You can get it in your legs and your arms. And it can be very protean, and it can be very different if you look at different people with the disorder. And so it is really important that we begin to try to understand this and to think about what we can do to move the needle. And so there's been a lot of work and a lot of collaborations, and there's been some alignment grants through Tyler's Hope. There's been several major granting efforts through Dystonia Medical Research Foundation. There's been the National Institutes of Health. And so everybody's kind of joining hands to try to push this forward. And so we have Edgar Rodriguez on, and this is somebody who's really experienced in the field of discovery and trying to push the envelope forward. So welcome, Edgar. And I want to ask you a few questions if that's okay with you. So I think you're still muted, Edgar.
Oh, I am muted.
There you go. You're back.
Can you hear me now?
Yes. So is it okay if we ask you some questions?
Yes, that would be just fine. Yeah, I'm actually just wanted to also take a brief moment to thank Rick and the staff family and Heather and everybody who's helped put this together. You know, I've been very, very fortunate to be able to work with Tyler's Hope. And so I'm really excited to be here and to help with whatever questions could have.
Oh, we lost you there.
Can you hear me now?
Yep, we got you now. Perfect.
Great.
Looks like the gaming headphones were not a good idea.
Yeah, no gaming today. So we have our live chat open, and I was going to ask you the first question, but you're getting a live chat from Reza Habibi, and Reza asks, what happens in the brain when someone gets dystonia, the mechanism? So what, you know, like in your mind, like as a scientist, you know, and this program today was sponsored by John Spence, and he's written a book called Awesomely Simple. And so I don't know if dystonia is awesomely simple. It's a great book, but like, this is a question, what's the awesomeness that is going on in the brain? What's causing this? Is it simple? Is it complex?
You know, that's actually, it's right on, very good, because it follows up with a statement that you made about DYT1 and you know, as a disease where people are focusing on. Primarily we focus on it, of course, because, you know, we have a clear genetic basis for it. So from a research perspective, it makes it somewhat simpler and cleaner for us to be able to model that in the research and be able to manipulate. That's what we're good at, right? Sort of asking questions, changing things left and right, and then observing what happens when we alter a number of things and make observations, and then try to reach conclusions and develop therapies. And the presence of mutations or you know, the clear link between a mutation and disease makes that process a lot simpler. And of course, you know, one of the things that we have learned amongst the many things is that, just like you referenced, even though the simple GAG deletion in that gene is what leads to DYT1 dystonia, What we have learned is that it is much more complex than that. And that just a simple deletion of 3 bases leads to all kinds of changes that appear to take place, even from early on in development, especially early on during development, and then through the life of the organism. And the mouse has really provided us with a good platform to be able to ask some of those questions. But I would say that for the most part, what we have learned, or I would say that there's two very important things that we have learned. One of them is that for the most part, it appears that neurons and most of the cells in the brain appear to stay somewhat relatively intact. And what I mean by that is, for example, when you compare it to Parkinson's disease or Alzheimer's disease, where you see a clear loss of cells throughout the entire brain, that's not necessarily the case in DYT1 dystonia. And so that's good, because it makes this not necessarily a neurodegenerative disease, but more like a neurodysfunctional disease. And like we like to say, if the players are still there in place, then there may be a way for us to try to fix them. And so the next thing that we've really learned out of this is that this is primarily a circuitry disease. It looks like, there's different, I guess I would like to call it departments within the brain. And, all those departments, just like in a company, they need to be able to talk to one another in a functional, effective way, right? You need your accounting department to be, pretty well communicated with marketing or with manufacturing in order to get the company to move forward. And I think what happens here is the brain, these different departments within the brain are not necessarily communicating with one another properly. And so that manifests itself in some level of dysfunction. So that's how I would put it from a simple research point of view. But of course, we'd love to hear your perspective on what you think is happening in the DYT1 dystonia brain.
Yeah. So, I mean, I would say in summary, what you said makes a lot of sense. That we have a situation, and we certainly call the brain cells neurons. So for people that are watching, the cells we call neurons, and those neurons are intact. They seem to be there. And so the difference between things like Alzheimer's and Parkinson and other diseases that we study that are degenerative, where the brain is sort of losing cells and wearing away, we're not seeing that in dystonia. At least that's not our understanding in DYT1 dystonia. And so somehow, you get an MRI, or you take a picture, you take a Kodak or whatever camera you have, and the brain looks pretty doggone good. From the outside, you say, well, what's going on? The brain looks good. So it's functional. There's something going on with the communication. So this small little deletion on 9th chromosome is leading to dysfunction, not only with the cells, but the pipes that are carrying the cells. And this abnormal conversation seems in some way to change the circuitry of what we call the basal ganglia, which is just the fancy term for that group of organs in the brain that are talking to each other, and it throws it out of balance. And if we record out of different areas of that quote basal ganglia, we'll see different recordings that are corresponding to a lot of the symptoms that we seem to be observing in these patients. And it's been a bit of a mystery, right? Because you can actually have the gene and not manifest any symptoms. It's sort of like, I like to say, if Mendel, who did the original crossing of peas and taught us how genetics worked, if Mendel was alive, he'd be turning over in his grave, because he wouldn't be able to figure out how this works. How can you cross the peas and then nothing happens? Or you cross the peas and you get one arm or one leg turns, or you cross the peas and you get the whole body or the face. And so it's very mysterious. And so, taken from that point of view, what I wanted to shift and ask you about is, okay, so if the chromosome, Edgar, that it's a deletion, and a little bit about the pathophysiology and where it is, is it in the nucleus, is it outside the nucleus, and what's going on we're starting to get a little bit of where the action is happening. What's the holdup with people developing therapies for DYT1? What are the key barriers that are keeping it from somebody from breaking through that ceiling?
Pat, so that's obviously a great question, and I think it's important to emphasize the point that you just made. For therapeutic development, is extremely important to understand the fact that indeed not everyone that inherits this deletion actually goes on to develop DYT1 dystonia, at least in the case of DYT1 for the GAG deletion. And I think that of itself is one thing that is always in the back of all the researchers that are looking into potential therapeutic development and potential therapies for this disease. I would say that, in the same way that we mentioned the fact that it is good that these cells are still remaining, that we don't really see this sort of overt loss of neurons like we do in Parkinson's and Alzheimer's. Yes, it makes it good for us from the perspective of, we still have these cells in there, so we have a shot at being able to fix them. But it also kind of masks for us, which are the cells that we actually need to target with a therapeutic, if that therapeutic requires to specifically target just those cells. And so, there's some new advances, actually. There's an amazing amount of work using mouse genetics, where we are just engineering all of these different mouse lines, and then we try to cross these lines to try to delineate precisely which are the cell types that are contributing to the disease the most, because that would provide us with a roadmap. And understanding that is important because now we know that molecular therapies in many ways have come of age, right? So now we can begin to talk about gene medicines like they are just, an aspirin in a bottle, right? We know that there are a number of genetic diseases that are now being treated, or number of clinical trials that have advanced, where we can take diseases where we know that there is a mutation in a specific gene, and if that mutation has resulted in the loss of the activity of that enzyme or the function of that protein, we can now try to replace that function. And there are a number of strategies to do that. At the University of Florida, of course, we pioneered the use of a virus that for the most part is non-pathogenic, pretty inert. And we utilize this virus called the adeno-associated virus, kind of like a FedEx truck or a UPS delivery guide, so that we can deliver a correct copy of a normal sequence of the gene or genetic information that is missing that underlies disease. And really, to be able to do that in the human brain is really almost like something out of a sci-fi novel. I mean, I'm still amazed as I think about kids today that are born with another horrible neurological disease called spinal muscular atrophy, can actually have access to a therapy which is based on this virus delivering a corrected copy of the gene that is missing in that specific disease. And so our hope, and the hope for a number of researchers, is that maybe we can do the same, at least in the case where there is a clear genetic underlying reason for the dystonia. So in the case of DYT1, we can try a number of strategies, right? We can try to, replace that gene with a normal copy. Again, that gene is called DYT1, but the protein that it makes is called torsin A. We know also now that torsin A has a family member called torsin B that is also able to come to the rescue. And so that's another strategy where we can try to augment the activity of torsin B to try to replace some of the dysfunction that is observed in DYT1. And there's a number of other strategies that I think we can talk as we move forward, including now the development, of course, of gene editing or genome editing technologies, which are hold all kinds of promise that are showing great efficacy already in humans. I think that is the sort of the next step for a number of neurological conditions. But again, our ability to deliver what I would like to call molecular medicine to potentially treat or cure dystonia is really will be guided by our ability to identify those cells and areas of the brain that will require the therapy.
So that's super interesting. And so you're saying that if you could load the gene product onto a virus, use that as the vehicle, you say the FedEx truck, but the vehicle to get it to the brain, that might be one strategy. Another strategy is this gene editing, and people have read about this in Time magazine, and and it's not just now scientists, lots of people are talking about this CRISPR type of pathway. There's some new CRISPRs too, like the CRISPR, it's like DOS 1, 2, 3 for those of you that are older enough to know what DOS was. So those are strategies. I mean, what do you think in terms of the next steps for those strategies? How far along do you think they are? I mean, are there significant experiments that need to happen in the dish and animal models, and what's holding them back from let's say a human trial? If we were talking in hypothetical terms, if everybody hears about this and reads about it on Time magazine, why not jump and do it now? What do people need to know? Why is it not moving faster in that direction? There must be barriers here.
Yeah, so I think it's pretty much, as we know, these are what we call biologicals or biological medicines, and there is a number of safety concerns that one needs to address. So when we think about the development of the therapies, there are two magic words. That's efficacy and safety. And you have to meet both of those things. And so typically the efficacy is it's the first step, right? You don't develop a drug or medicine unless it has some sort of efficacy in a model system. And again, going back to the DYT1 scenario, our ability to manipulate that gene in the mouse allows us to establish models that then can give us a readout for the efficacy of a number of these approaches. And so the approach, for example, that I discussed regarding the augmentation of either Torsin A expression or Torsin B, from an efficacy perspective, requires us to understand, how much of this gene do we have to deliver, and how much do we have to express in order to see a therapeutic signal in the model. And then from a safety perspective, we need to make sure we understand, do we have to control how much of that gene, right? So it's not as easy as just saying, just give it more Torsin A or give it more Torsin B. We have to understand the titration or the levels, how much of this gene, and do we have to sort of selectively limit where that gene will be expressed, what cell type will actually express that new therapeutic gene. And so it takes time. Some of these studies, some of these models don't really show any dystonia-like symptoms, for months. So sometimes, you do the treatment, then you have to wait a few months to be able to finish your experiment. You have to analyze all that data. And so we're just now at the cusp of really trying to identify exactly which genetic cargoes are going to be the best for us to be able to move forward. And so we're really in the phase of drug development that is called discovery. We're still at that discovery phase. And so what does it take? It takes, as we all know, some amount of resources. And it's not just financial resources. Of course, money is always important, but the sort of the human resource is a key here. Having as many researchers involved in this problem and tackling the question is extremely important because it provides a diversity of approaches. And I think that's one goal of mine as a science advisor at Tyler's Hope, is to try to continue to recruit as many researchers to get involved in this question as possible, and to try to move them, try to identify what resources are available and try to connect them to those resources, establish collaborations that will move some of these problems and some of these investigations forward as quick as possible. And I think, that's one thing that we've been able to achieve. And if I could just take 1 minute, for example, if I can highlight, we have been extremely involved in identifying groups that will work on the question of genome editing for dystonias, specifically for DYT1. And really by providing them with some mechanisms by which these groups can come together and work together, even though they're in different institutions, they can then collaborate, bring their own expertise, and then try to move forward, with the therapeutic. And recently, actually, some of those efforts were recently published at a journal of molecular therapy, showing that CRISPR-Cas9 systems can be utilized to selectively target the allele or the mutant allele, mutant copy of the DY21 allele in human cells. And I think that's great, not just the aspect of the therapeutic itself, but the fact that there is a possibility for us to target precisely just the mutated allele in a human cell. And so it opens up a whole new avenue of potential therapeutics directed at the DNA level. So why does it take this long? Again, these are complex therapies. They're not just simple molecules. And so these complex therapies require a lot of fine tuning. That being said, I'm really happy to see that Tyler's Hope has also been involved in trying to push forward in the area of small molecule development. And I think that's also another area where, you want as many shots on goal as you can. And that's an area where we know there's a clear path. We have many decades of experience on how to develop small molecule compounds that can penetrate the brain and that can act in targets and provide good target engagement in the brain. And so I think those efforts are also important for us to be pushing ahead. So it's not just about the molecular medicines, but also small molecules pushing ahead.
Yeah, that's great. I want to remind everybody we have a chat open here, and I notice we've got John Teller and Janet Hirschedler. I see you, King Lee, and a whole bunch of really great people in dystonia and partners and friends. And so feel free to jump in here on the chat as well. And I want to shift a little bit, Edgar, and ask you a little bit about medicines. And one of the things that I know from having a few decades of experience with patients with dystonia is that there's a frustration in the medicines. One of the best medicines, if not the best medicine we have, causes a lot of side effects for patients with dystonia. So it becomes a dose limiting issue for people, and particularly can affect school performance in kids. That blocks these receptors in the brain called cholinergic receptors. But could be important to understanding some of even the pathophysiology or how these different cells and pipes are all working together. So what is it? So you've commented now on, you load up the virus and you send the virus, and there's some potential for gene editing and those types of technologies. Tell me why, what in your mind from a scientific advisory standpoint, tell me a little bit about the barriers of why we haven't been able to get specific drugs in there. And I know there's been several partnerships between numbers of different people, the National Institutes of Health, DMRF, Tyler's Hope, and tell me what are the barriers? And I've lived long enough through, like we had purple compound and all these different drugs that were going to cure DYT1 dystonia. What's the, the antibiotics, of course, anti-malarial drugs. Anybody who's been around medicine a long time knows that when anti-malarial drugs came up for corona, those of us who have been around the block laughed because they come up on a lot of drug screens for a lot of diseases. So it wasn't, it certainly needed a little bit more attention. But, why? Tell us a little bit about what do you think the barriers are here to is it small molecule development? Is it specificity? Is it that we don't understand the disease well enough? I mean,
Yeah.
what do you think is it? Because we certainly got a lot of committed partners here. Nicole Kalakos is working on hits now. So where do we need to go? Where do we need to invest our money?
That's exactly right. I mean, when it comes to small molecule development, as you well know, and as we all know, it just requires a lot of screening. And just like any other screening, you need to have a good model system. What is it that you're screening for? You have to understand the pathophysiology of the disease. You have to have a model system. The model system has to be simple enough to fit in a very, very tiny well. As you know, a lot of these small molecule screens are, thousands and thousands and thousands of compounds that have to be done, in triplicates. They have to be done with some, negative controls. They have to be focused on molecules that can actually cross the brain. As we know, thankfully, our brain is pretty well protected. And so, anytime we ingest things, our brain is designed to try to keep those things that it doesn't want out. And so it's important for us that when we design these small molecules, that these molecules have the ability to penetrate the brain at the doses in which they're going to actually engage their target. And so it's almost like playing hide and seek. And that's the simple scenario when you actually know your target. As we have discussed, we still have so many questions about DYTY and dystonia. We know that there is a dysfunction of these neurons. We know that they're not communicating properly, but why? Why aren't they communicating properly? In the case of DYT1, we know that the gene has some abnormal folding kinetic behaviors, but how does that lead to neurons not functioning properly? And why only neurons? And why is it that the manifestation is at the circuitry level? So there's all these questions that are still part of our lack of understanding the pathophysiology of the disease that preclude us from being very narrowed and focused in our screening of these small molecules. Now, that being said, scientists, as you know, we're stubborn, right? We're not going to stop just because, our models may not be perfect. We're going to keep going. We understand that in our profession, 9 out of 10 things go wrong. And so we have to keep hitting and hitting until we find that one. And you mentioned Nicole Kalakos from North Carolina, who's doing amazing work looking at compounds that, small molecules that could impact the circuitry function in the brain. And so sometimes, as you know, it's important for you to know the mechanism of action, right? To understand a target and to understand how your small molecule engages that target to modify the disease. But sometimes you get lucky and you get an effect. You don't necessarily know how that effect is working, but if it does work and if it's safe and it's showing efficacy, we can figure out the mechanism of action later. Let's just get that drug in the clinic and into patients. And so, you battle with those two fronts, right? When we were discussing biologics, in the case of biologics, you know exactly what you're targeting, right? Those biologics are designed to be precise in their mechanism of action. In many ways, you're still guessing that mechanism of action is actually involved in an important aspect of the disease. But a lot of small drugs and a lot of molecules, just like you alluded to, these antimalarial drugs, you're just looking for a signal and outcome. And you may think they're acting on a specific target, but in reality, the effect may be driven by something else. So specificity doesn't tend to be a characteristic that is associated with small molecules. But as long as they're efficacious and safe, they're moved onto the clinic. And so, rest assured that if there are molecules that have been identified that work well in model systems, those molecules are going to be moved forward into the clinic as long as they can penetrate the brain, where we know the effect or the disease causing pathophysiology is located. And as long as they don't cause any toxicities in peripheral organs. That's one of the barriers to small molecule development, is that oftentimes you find yourself having to dose the patients so high with that small molecule in order to engage those targets in the brain, that then peripheral organs begin to suffer some consequences, or what we like to call adverse events, that then can have an impact in the health of the patient, independent of whatever effect they're having on the dystonia symptoms.
Yeah. So I think that's, really a brilliant answer and super important. And turning over to the chat here, we have a question from Richard Staub about what can we do besides fund research to have a bigger voice or impact to find a cure? Well, one thing I would say is recently Ray Dorsey at Rochester and Bas Bloem in Nijmegen in the Netherlands, and Todd Scherer, who's at the Michael J. Fox Foundation, we wrote this book. And while we were writing this book, we did a lot of research. We looked into polio and HIV and what did it take to end those diseases, or at least end the tyranny of those diseases. And it occurred to us that there is a formula, and we write a lot about this formula, and it applies to Parkinson, it applies to dystonia, and we call it the PACT, the pillars: prevent, advocate, care, and treat. And in treat, developing new treatments. And the things that we noticed that were important to making a big impact, well, if you look at, let's say, HIV, once we started putting in $3 billion a year with a B into research, the needle moved. If you look at Parkinson's disease at the NIH, we're putting in 200 to 300 million, maybe 290 to 300 million, depending on the year. If you look at dystonia, we can look that up, but it's going to be, very low. I'm going to guess it's 20 million or less. Probably Jan or someone who's looked at this more recently with some expertise at DMRF could tell you, but the numbers are way low. So we need a log scale of funding to move the needle. That's one thing. We need to prevent, advocate, care, develop treatments. And then we need to be more charismatic. So when we turn the tides in HIV, people went and put a condom on the house of the Congressman Helms, from South Carolina's house to make their point. They occupied the FDA, they occupied the federal mall. We need to be more charismatic. We need to be more aggressive. We're not moving the needle enough. And I think as a group, we're going to need to do those sorts of things to move the needle. People are also asking about staying busy during the time. And, I people who know me like to know that I don't like to preach at patients. I like to say we're like your cabinet advisors. We give you advice. And if you do something we tell you not to do, that's okay. Just tell us what you did, because it might have worked, and we can't learn unless you tell us, right? So we try not to be paternalistic. But I will tell you, during this corona virus COVID-19 epidemic, we have become paternalistic, and we need to be paternalistic, because if we don't educate people, we're not going to save lives. And there are lives here at stake with a very high mortality, particularly in elderly and in underserved populations. And in the elderly population, as much as 1 in 5 with the mortality, particularly if there's a need to ventilate. So we need to keep people at home as much as possible. We need people to understand that we're serious about covering the face, and it is mostly a respiratory disease, and the countries that are covering their faces makes a big difference. And so we need to tell our dystonia patients not to cave to peer pressure. When you see people in restaurants or other things that aren't doing it, you need to do it. You need to stay safe and make sure if people that you don't know aren't safe, you need to be distanced from them. You need to have your face covered. They need to have their face covered, and not time to eat or drink. And lay low like a mobster until this thing is done. You gotta be smart. Now, one other thing, and there's a question that I have in front of me that was emailed in, and that's about, do you have a higher risk of dystonia? If you have dystonia, do you have a higher risk of getting corona? We actually don't have that data. Well, we don't believe that's true for a lot of diseases like dystonia or Parkinson or Tourette, that it's a higher risk because you have that you're going to get corona or COVID-19. But if you do get it, if you have a comorbid another disease, it may be more difficult to recover. And particularly if your dystonia affects your respiratory muscles or your other muscles, you've already got, something that's making your life difficult, might be, affecting your ability to walk. And so if you add something to it, that makes recovery more difficult. And so it's going to take really, a long time for us to get this data out. By the time we have the data to know about the risks, we'll have things under control. So we need to take as much as we know now and try to move forward with a strategy that's going to save as many people as possible. And so that's really face coverings and being disciplined. Now, in terms of the advice of what to do when you're bored at home, I've seen several questions on this. Edgar, any advice for our group here? If you have dystonia and you're at home and you've got cabin fever, what do you do? What do you help?
Unfortunately, as you know, we scientists are not, quite well versed on fun. But I can only say what I have done myself, right? So, I've tried my hardest to spend some time outside whenever possible. Oftentimes our lives are lived from office to office or building to building. So I've actually, dug into the dirt, moved plants around, do a few things that require me to interact with nature a bit more. I've done some reading, and quite frankly and honestly, I've done a lot of movie watching, which I haven't done in a long time. So it's been good. Actually, I have a son who's 14, and there's a few movies from the 80s that I remember watching when I was his age. And, so it's been good to share those with him as well. So you get some time to be able to spend with family, and you try your best to enhance some of the things that we know are good for our health, but we don't really spend too much time doing. And so now that we have the time and we're required to have to stay home, then I think, it's a good time to enjoy those. And one thing I wanted to point out also, you were talking about coronavirus, and of course, we've talked, everybody's heard so much about testing and coronavirus testing. And one question or a few questions actually that I saw that came through have to do with genetic testing in the case of dystonias. And, I wanted to ask you, Michael, what your thoughts were on, the availability of these tests. And, do you see now in the clinic a move towards being able to test as many patients as possible for genetic underlying pinnings for some of these diseases? As you know, having that genetic information is key for us to be able to push therapeutics forward. So I'm wondering, what the status is of that in the clinic.
Yeah, so it's a great question. We're seeing all sorts of shifts, across, at least across my career. So we're now seeing a shift where people, for example, are interested now in trying cannabinoids for the brain. And 20 years ago, if you said cannabinoids and taking marijuana or stimulating in the brain, people would have said you're crazy. And now people say, well, it's great for anxiety and sleep and pain, and has helped people with dystonia and other disorders. And genetics is the same deal, right? We, all the classic people that have been there have said you need a genetic counselor and you got to be careful and you don't want to do this. And it hasn't made it to the Supreme Court, but it's clear that opinion is shifting, that people feel that this is, their DNA is their DNA and they want to know. But not everybody. And we know that going all the way back to to the writings of many people, particularly in the early 80s when Huntington's disease was described by Jim Casella at Harvard and by Nancy and her group that went to Maracaibo and the people that were at risk. We learned not everybody wants to know. So having said that, it's sort of changing, and a lot of people are getting genetic tests, and we're getting so much information, we're not sure what to do with the information. Sometimes you have a gene, and remember I told you Mendel turns it over in his grave, because if you get the DYT1 gene, you don't even know, like, if you're even going to get symptoms, much less what kind of symptoms. We don't know why. And somebody asked a question about that. I think Ann asked that question. We still don't know why, Ann. I mean, like, there's a lot of speculation, but I think anybody that tells you they know, exactly why is still maybe fibbing a little bit on that side. So on the genetic side, we're now being humbled. So we're uncovering all sorts of mutations. And so from a group that I got the pleasure of working in my career with, Kelly Foote, and he's done hundreds of patients with dystonia, DBS, and we've seen all these interesting kids and adults, and we didn't know what they had. And it's recently, as people are getting genetic tests, they come back and go, oh, you have this. And we didn't know that, at the time. We just knew they had a phenotype that, seemed to be responding, and we were trying to understand what was responding and what wasn't. And so it is giving us a sense of the pathophysiology. What do those genes do, and what pathways are they? But we also have to be careful, because a lot of times we come up with genes and it doesn't mean that it's causing the disease. And so, we have to be really careful. So right now we're spun a little bit out of control, like we're a little bit undisciplined. We still don't know. We don't know what we don't know. And I like to say every day that I practice medicine, I know less. I mean, I know less today than I knew yesterday. The more I learn, the more I realize I know less. And so you get humbled on these things. So people are going out and they're getting panels from companies like Invitae or many others. I mean, that's not the only one. And 23andMe has a group, and Athena Diagnostics and Mayo Clinic, and so everybody has a different way of doing the genes, and depending on who does the genes or what technique, and this as a scientist, it may come up positive or negative. Some of the genes, like dopa responsive dystonia, have so many different mutations, you can't even develop a panel for them. And so it is information overload, and it's blowing the minds of all the classic neurologists. They all need to retire now, because they used to say, okay, if you see this, then it's this disease. If you don't see this, it's this disease. And now there's so many exceptions that they're like, I better just retire and put my bag away, because now with genetics, I can't figure it out. So it has become a really tricky area. And if it's confusing and tricky for us that know some of the science, imagine how tricky it is for the family. So I think on the major mutations, like knowing you have DYT1 positive is helpful for prognosis and for picking treatments, like maybe DBS and other things. And then the other common mutation in adult onset, sometimes focal dystonia, sometimes more than focal, this DYT6 or THAP1 mutation is very common. And so those two, are really dominating the discussion, but others are quickly emerging. And then there's all sorts of modifiers. And as I think Ann was asking, what turns these on and off, and the environmental influence. So it's very confusing. Now, would I spend thousands of dollars to get a genetic test? Probably not. A lot of these companies have gotten much cheaper, so if you talk to your doctors, a lot of these companies in the market are really getting very competitive, and it's no longer necessarily a $5,000 or $6,000 sum game, or you don't have to send it to a great researcher like Matt Farrer, or to Sue Bressman or Lori Ozelius, who are just magnificent at dystonia genes. A lot of these you can get, available through other methods, but it doesn't necessarily yet change the approach to treatment. It hasn't, the needle, you're getting close in a couple of things, but the needle hasn't shifted yet. But we do think, and people are asking what's going to happen in the next 5 or 10 years. We do think that it is soon going to be important in neurological diseases, like it is in cancer, to know your genotype if you have one, because knowing your genotype then let somebody like Edgar, attack that specific genotype with a very precise therapy. And so it may not be a one size fits all. It may not be the granola bar that everybody eats and gets better from dystonia, but understanding some of these relationships could be important. So,
as
the longer the answer, that means I don't know the answer, right? If I keep talking. So, but Anyway, Edgar, what
No,
were
I
you
was
going to
just
say?
gonna say, I mean, you hit right on there at the end, right? The phrase personalized medicine is something that now is becoming almost streamlined, right? And it seems, primarily be associated with your genetic makeup. And so, there's obviously whole new areas of pharmacogenomics, which is not just identifying, mutations or sequence changes that are impacting a disease, but actually sequences within your own DNA that impact whether you respond to a treatment or not. And they, sequences within your genome that determine how well you're going to metabolize a drug when you're given the drug. And so there's all this whole, this entire makeup of questions and aspects regarding genetics that I think is starting to be important in medicine. I think it was Elon Musk recently stated that it's good that we're moving so much to DNA and RNA-based drugs, because then it shifts the paradigm to a software problem. And of course, that's a little too optimistic. But then again, he's never been accused of being a negative kind of guy or a pessimistic kind of guy. And so, in many ways, it's somewhat true, right? The idea that if we can pinpoint, phenotypes, or what we like to call, either diseases or how people react to a medicine, if we can make associations between that and a genetic makeup, then really that provides a pretty strong, powerful scenario for us to be able to develop therapies for individuals. One, I know we're coming up in an hour here, and so we'll probably be, starting to close pretty soon. But there's one question that I would like to tackle that I think highlights some of the difficulties and challenges that we face in the lab. And the question was relating to a comment that I had made earlier about the fact that sometimes these mouse models take time to develop, the sort of signals or disease manifestations that we're trying to measure. And so the question is, can you expedite that? Can you make these models, develop these symptoms faster so that you can screen faster and do things faster? Trust us, that's exactly what we all try to do. But one of the drawbacks with that, where you have to be balanced, is you can make the model go faster, but that could preclude you from being able to really ascertain properly whether your therapy could work, or whether your therapy can translate to the human condition. Because sometimes when you try to push these systems to go really fast, then all kinds of other things start happening, and what we like to call that black box. And then your therapy, trial may give you a false positive, or worse yet, may give you a false negative. And that is all because the model is really out of time with what the actual sequence of events are that underlie the disease in humans. So you have to kind of play that game of being able to develop models that are quick enough so that they're useful, but yet have sort of the spatial temporal components that are key to the disease, so that when you're developing your therapies, you get a better signal and you're more confident moving that drug forward to the clinic.
Yeah, so that's terrific. And just as a last comment, I'll say that we do have genetics counselors in multiple of the centers, that work along with Tyler's Hope and DMRF and other organizations. Ours is named Lee Kugelman, who joined us from Emory, and she can help with counseling. But a lot of people, before they get genetics panels, are going to want to get counseling and decide whether they want to actually get it. So I don't want everybody to go out and just order a panel, because it can change your life knowing your genetic status, and we may not know how to explain that panel. So I think genetics counselors can be super important with that. And we also continue in pushing the needle to try to work with state governments. And so Rick Staub and Tyler Hope have been really just amazing work with the state governments to provide funding into dystonia research. And as Edgar said, we need more people. We have a critical mass problem for researchers. We need to train more people. We need to have more fellowships. And so it's super important. And since we didn't get to all your questions here and we're coming right up, we just hit the hour mark. We're going to pause here, hand it back to Heather, and maybe we'll be able to convince Dr. Foote to come on and talk about DBS, since we had so many deep brain stimulation questions, and maybe Dr. Shukla, one of the experts, talk about some of the other Botox and dystonia and treatments, and maybe somebody will come talk about medicines and Adolfo, or maybe Lori Ozelius or Matt Fair, or somebody can talk about genetics. But these are all really interesting topics, and we thank you for spending the hour with us, and thanks for having us, to Tyler's Hope, and thanks to John Spence. And I'll just say, John, your book is awesomely simple, and I hope that that's how DYT1 chromosome 9 ends, as an awesomely simple exercise. But right now it's gotten a little complex. So Heather, back to you.
All right, thank you, everybody. I am going to go ahead and just share this slide with you. Like I've said, and I'll say it again, if you guys have any questions or I can do anything for you, please feel free to email me. If you have any more questions, we're going to try and get all these questions answered. If you asked a question anonymously, if you shoot me an email and let me know who asked the question, I'm going to try and get those answered and sent back to you as timely as possible. And we kind of hit a little bit on the registry, the Dystonia International Patient Registry. This registry was created to give as much information to researchers as possible. If you are interested in registering, you can go on to the Tyler's Hope site. I also sent out a link to sign up when you got the email for the webinar. But again, if you have any questions, please feel free to reach out. And thank you to Dr. Oaken. Thank you to Dr. Rodriguez. Thank you to John Spence and his staff for your help. And we really appreciate your time.
Thank you, everyone, for your time. Have a good evening.
Thank you. It was fun.